Okay, our second presentation is by Lynae Brayboy, and Lynae's a faculty member at Brown University, and she's going to speak on designing research to inform medical practice. Lynae.
Good morning. Thank you very much. It's an honor to be here. I'm delighted to actually speak about designing research to inform medical practice. And so I want to make sure I know how to use this. Green is advance, and this is for the light. Okay, so first I'm going to tell you a story. I am a physician scientist, and I entered the field of reproductive toxicology because I was interested in a subspecialty of reproductive endocrinology and infertility, and that's oncofertility. So we actually preserve the fertility of women who will be subjected to some type of gonatotoxic agent. And so that's typically women who have been diagnosed with cancer. And so this is really important to me because a lot of women will be diagnosed with cancer, and many women, as you see here, will have breast cancer. And many, many women will actually survive their breast cancer, about 90% at all stages on average. And so they will undergo a chemotherapy that will save their lives, but will damage their ovaries. And they actually use many, many alkylating agents to do that. And what we can do is actually we can preserve their fertility. And the way we used to do it a long time ago, we used to give GnRH agonists. And what that does, we thought, was to quiet the hypothalamic pituitary ovarian axis. And actually, we found that that's actually not that effective. And one thing that we could do instead is actually do embryo cryopreservation. So we've been doing that for years and years. However, we actually, in the last 4 to 5 years, since I finished fellowship training, we found that we can actually preserve oocytes. And so that's the standard of care right now in our field. And that's great. That's wonderful. If you live here, you're in good shape. We can help you. We can cryopreserve your eggs. We can cryopreserve your embryos. And that's wonderful. But since I used to live here in sub-Saharan Africa at some point, that's a problem. And what do we do for you? We don't have a lot of things that we can actually do for you. And fertility is really just one thing that the ovary provides for you. There are many other things that the ovary does, actually. It makes estrogens and androgens, and those both have an impact on many different functions. I want to point out that women who get alkylating and chemotherapy are susceptible or prone to sexual dysfunction, vasomotor symptoms, which are commonly known as hot flushes or hot flashes, and they also have increased cardiovascular risk and risk of osteoporosis. And so it can really affect the quality of life after they've already survived cancer. So I entered the field of reproductive toxicology because I was interested in doing much more than just preserving the fertility of these women, but how can we preserve their ovarian function? And so reproductive toxicology is much more than chemotherapy. There's many more things that are included in that. And so there's a whole different subset of fields, and this is a slide from NIEHS, and I specifically concentrate, oops, it's a little bit lagged, there we go. I specifically concentrate in reproductive medicine in the ovary. And so I'm starting to get interested in transgenerational consequences of exposure that are non-gestational. And so one of the issues is that there's chemotherapy, but a lot of chemotherapies have been derived from nitrogen mustard. And nitrogen mustard has been around for a very long time, but unfortunately, we don't know much about its transgenerational effects. And nitrogen mustard is actually a mutagen that's a DNA alkylating agent, causes adducts. It's been around since World War I, and it's been used on soldiers in the battlefield for a very long time. And unfortunately, it's fallen into some bad hands. And so it could be used on a large scale, and it has been reported to be used in Iraq and Iran. And so we know that women who've served in our military have potentially been exposed to this, and women make up about 12 to 15% of all of our US veterans. And that's problematic to me because it's an occupational exposure they're maybe not aware of. And as a woman in the military, they will pass on potentially mitochondria that have been either destroyed or potentially hurt by nitrogen mustard exposure. And so we're just getting into looking at how nitrogen mustard affects the mitochondria in the oocyte. And let me point out that it's very important that there are a lot of mitochondria actually in a mature oocyte, about 100,000. And it's the rarest cell in the body, but it has the most mitochondria. And that's important because the oocyte has to be fertilized, and then it has to transit from the ampulla to the endometrium and implant. And during that time, there's no blood supply. So it has to rely on those mitochondria to implant and do placentation effectively. So we just have some preliminary data that shows that maybe oocytes that are exposed to in vitro nitrogen mustard have abnormal bioenergetics. One limitation working with oocytes is that you need a lot of oocytes to do assays like this. You need about 100 oocytes, and that represents about 30 sacrifice of mice. And so it's very expensive to do types of research like this. And you can't get replicates at the same time because it's just technically not feasible. So another thing that we've looked at is actually a reactive oxygen species. And we've looked at dosages that are used clinically because nitrogen mustard is still used as a chemotherapeutic. And we've seen, and the picture is not showing up lovely here, but oops. We've seen here that at dosages here that we have increased ROS in the oocytes. And remember, these are the oocytes, these are germinal vesicle oocytes that are immature, that are in the ovary, and that will be exposed for the duration of the woman's life. So this is really quite important to us. And then when we look at electron microscopy, we see that the oocytes of the normal oocytes that are controls, we see that their mitochondria have normal forms. But when we look at the nitrogen mustard treated, we're concerned that maybe the mitochondria have been affected. And so how do we best study this? And so that's one of my current projects that we're still trying to figure out to this day. We think that also oocytes have been reported in the literature to not undergo mitophagy, which is autophagy of the mitochondria. And so if this is not occurring, they're being passed on to the next generation. And that since they're maternally inherited, this is very deleterious for the embryo. And we think that maybe in our oocytes, in our hands, that we do see lysosomes, which are pictured here, which is a part of type 3 mitophagy. And we're still confirming this. So switching gears now, I wondered also too about not only the molecular functions, but also the long-term reproductive implications. One of the problems that I've had in the clinic is that when patients get chemotherapy, they ask, well, if I have children after this, what happens to my children? And I don't have an answer for them. And so I've actually used my problems in the clinic to design clinical research or basic science research that would help impact clinical outcomes. And so we're going to look at the F1 and F2 generations, at their ovaries, the mitochondria in their oocytes, and also their reproductive characteristics when they achieve sexual reproduction and how many pups are in the litter, and so on and so forth. And so we know that alkylating agents are very toxic, but I always wondered, does the ovary have some capacity to protect itself from any toxicant, not just alkylating agents? And so I am in a starfish lab, and in that lab, I was able to read some literature about a transporter that's in the plasma membrane called multi-drug resistance transporter. And so in starfish oocytes, they're present. And so that makes sense because starfish spawn their eggs into the ocean, and they're in pollution, they're in different salinities, and they have to be able to efflux toxicants. And so I started to look at MDRs, and again, they are ATP-driven pumps that will bind ATP to efflux a ligand. They efflux about 4,000 different compounds, and so they are called promiscuous in the literature because they can efflux many different things. And the other thing is that there's so many, but 49 are actually have been reported in the human body and in the reproductive tract, and we think about 23 of those effluxers are actually in the ovary. So they seem important. And the other thing I want to just tell you is that they actually have an estrogen response element. And what does the ovary make but estrogen? And so they seem very, very important. So the more I looked at them, the more I thought, this could be interesting. So I will just reference MDR1 as P-glycoprotein and ABCB1 interchangeably. So for my fellowship project, so in reproductive endocrinology, we spend 18 months in the clinic and 18 months in at the lab. And 18 months in the clinic, we learn how to do in vitro fertilization, we learn how to do embryo transfers, and we understand how to do superovulation. But in the lab, it's kind of an open-ended thing. And so I thought, well, I'm interested in MDRs. Let me see if they're in mammalian oocytes. And so it turns out, lo and behold, they are in mammalian oocytes, and they're expressed at all of the maturational stages. So in the GV, that would be in the ovary, and these are oocytes that are preparing for ovulation, and this is an ovulatory oocyte. So they're there and they're present. So I thought, that's great, but are they functional? So what if we took cyclophosphamide and we incubated these oocytes from mice with cyclophosphamide? Will you get any cell death? And it turns out, though they're alive, they're here, this is calcine AM. So if a cell is alive and it has intact esterase activity, it'll cleave acetomethoxy acetate, and you'll see this green fluorescence here. But if it's not viable, the plasma membrane will have been affected or impacted by something, and ethidium homodimer 1 will be able to come in and bind to nucleic acids and cause it to fluoresce red. And if we see that, we think, okay, the cell is not viable. When we inhibited MDR1 with an agent called PSC833, we saw that loss of viability in all of our mouse oocytes as well as human oocytes. So I used discarded human oocytes from cycles that have not been fertilized, and what I do is I did the same assay here. So I thought, okay, they're there and they're functional. They're protecting the oocyte. What about in the entire ovary? So after I finished fellowship, I decided to apply for a K award, and that K award was really mainly to investigate MDRs in the entire ovary. Because again, like I said, oocytes are the rarest cell in the body, and so the ovary is really mostly somatic cells that are protecting and supporting that oocyte. And so when we look here, that there's definitely MDR in the wild type ovary, and this is the brown staining that you see throughout the ovary. This is a cross-section of the ovary. And you see here in the knockout, we definitely have less expression, although it's not completely gone. There could be other MDRs that the antibody is actually binding to. So here, we also see in the knockouts versus the wild type that there's more cell death when you actually treat the animals with cyclophosphamide. So not having MDR makes your ovary vulnerable to toxicity. And so that was really impactful for me. The same thing that we found in the oocyte, now we're seeing in all the somatic cells. When we looked at the ovaries more carefully of these knockout animals, and these animals in particular were missing MDR1A and B, it's 2 genes in the mouse, and BCRP, which is another MDR transporter, which was mentioned yesterday in a talk, we saw that they actually have different folliculogenesis. So they actually have more preantral follicles and less corpora lutea. And what does that mean? Well, they're actually getting more follicles to maturation, but then they're not ovulating them. So maybe there's inherently something wrong with these ovaries. They haven't had a toxic insult, but they're having this abnormal folliculogenesis, and that's problematic, and that could be manifested as infertility potentially in a human. But mice are very reproductively fit, so that's not the case here. So when we looked at the metabolites of those knockouts, we saw some abnormalities, and what I'm projecting here is a mutant, but I just want to tell you about the knockouts abnormalities. They actually had reduced glutathione and other metabolites consistent with oxidative stress. But that's a knockout, and that may not actually represent what's happening in a human. Humans are more likely to obviously have a mutation. And so we wanted to look at the spontaneous mutant that was available, and this is an MDR1 a mutant that is found about 25% of the time in a CF1 background. When we looked at those ovaries, we found that the metabolites that were accumulated were metabolites that were all involved in the TCA cycle. So now I'm going back to mitochondria, and I think, okay, so these metabolites are all important to the TCA cycle. Could this be important, and are MDRs and mitochondria linked? So again, we wanted to just test, do they have that same phenotype that we saw in the knockout animals? And when we blocked MDR1 with PSC833, and turns out they do at 48 hours, we do see increased susceptibility. So again, they're vulnerable and their metabolites are abnormal. We looked at the oocytes themselves to see if they had more ROS, and it turns out that they do. And these, again, are the GV oocytes that are present in the ovary for the duration of the reproductive lifespan of the animal. And when we looked at the transcriptome of these animals compared to their wild-type litter mates, we found some interesting differences. So this is a bicluster graph, and you may not be able to see too well, but there's several mitochondrial genes that are implicated in complex 1. And so it seems like not only is MDR there to protect the ovary and protect the oocyte, perhaps there's a link between MDR1 and the mitochondria, and that link could be important at protecting the entire ovary and the oocyte from actually everyday toxicants. And those toxicants could potentially lead to a cancer phenotype or oncocytic phenotype, which has been described in the literature. So all in all, I've tried to teach myself how to do IPA analysis, and I'm still learning. But when I looked at those differentially expressed genes, I looked at some of the diseases that they map to. and they tended to be mitochondrial disease as well as some diseases involved in reproductive development. And so we're still looking at these. So some of my ideas to how we can incorporate gene editing tools is, I have a couple, and I would definitely like feedback, but one of the things that I'd like to do in the future is actually do a knock-in of MDR1, and that would maybe be a temporary expression so that we could actually prevent toxic exposure and protect the ovary. Another thing is to use gene editing tools to actually incorporate SNPs that we know are expressed in humans, and there are several SNPs that have been reported to being associated with increased lung cancer and increased breast cancer, and potentially that's because there's an inflammatory state. And so that's, I think, a really exciting idea. And listening to this workshop, I have hope that maybe we can do that in a tissue-specific way. So also a tissue-specific knockout of MDR1 to test different toxicants in the ovary would be very exciting as well to do. So this has already been done in zebrafish, and what they did is they used a tissue-specific promoter. And so it seems like it's possible to do. Not sure yet how we would do it in a mouse, and I invite your ideas and collaboration. And I just briefly wanted to mention the fact that we didn't talk about this today, and it's been kind of the pink elephant in the room, is that CRISPR and gene editing tools in the human is obviously still a very long ways away, but I wanted to just give you an idea of what's been published in our literature, and that it's still a very big bioethical dilemma, but it is an alternative to what we do, which is pre-implantation genetic diagnosis. And what that means is that we actually assay an embryo to see if it's euploid and genetically normal if we know that there's a genetic disease that's in question. But this seems still a very far away to do. And also there's the question of mosaicism, which we discussed yesterday, and also off-target effects. And eggs and sperm are easily manipulated, so it's something that would be feasible to do, but yet we still need to do a lot of work around this. So I'd like to thank everybody for your attention. I'd be happy to take questions. Sorry, I was going rapid fire too, to try to get on time.
Okay, if not, I think we'll go ahead and move into the panel discussion. And so